Hello, today we're going to talk about restriction enzymes in gel electrophoresis. This is such an amazing technology that has been discovered and worked through that you're going to absolutely love it. First of all, a restriction enzyme is like a molecular scissor or a scalpel. And what it does is it cuts DNA in what we call a precise and predictable manner. So it's very specific on how it will cut the DNA. It will actually break what are called phosphodiester bonds that hold the nucleotides together. So it breaks them apart at the nucleotides, not the hydrogen bonds between the bases. So you can see here it just kind of breaks it. Okay. There are two main types of restriction enzymes. We have endonucleases, which cut DNA internally. So think of internal. And then we have exonucleotides that cut them at the ends or the exit sides. And they are named quite crazy. Just to give you an idea, we have EcoR1, we have BAMH1, and HIND3. So the E refers to the genus for the bacteria E. coli. The Co is the species for coli, so this is coming from the bacteria E. coli. R is the strain, so E. coli with the strain RY13. And the 1 is that it's the first endonuclease isolated. And again, endonuclease is internal. Okay, this is just showing you what EcoR would look like. Okay, so we said that restriction enzymes cut DNA in precise and predictable manners. Well, what they do is they look for palindrome sites. Okay, and this is a symmetrical nucleotide sequence between the two strands of DNA. So if you look here, it goes GAATTC, and if you remember, they're anti-parallel, so it's read in the same way, GAATTC. So these is called a palindrome. It's read in one direction is the same as it read in the other direction. So our restriction site is the actual place that the DNA is cut and the palindrome. So for the GAATTC palindrome, it's actually going to cut it between the G and the A. So it makes this cut. So you get this. This area right here and this is called the sticky end because they are free bases. Just showing you again how it's cutting. So once we can cut DNA up into little fragments, we can then visualize it by running it on a gel or performing gel electrophoresis. So just so you know, DNA is negatively charged. So when you plug in your little apparatus, the DNA will travel to the positive pole because you're plugging it into a power supply. That is going to allow the DNA fragments to sort by size, and that is because it's found in this polyacrylamide gel, and you'll get to see this in the lab. So DNA is loaded here at the top and these wells. Okay, there's a power source. You plug it in and the DNA will travel towards the positive end, sorting the bases or the fragments out by size. So the larger bases are on are on top, and the smaller fragments are on the bottom. This is just showing you what it would look like. This is showing you an actual rape case. You always run what are called ladders, and those are just kind of standard so you have a basis for comparison. Okay, so this is the victim, and there's two suspects. This suspect has 2 bands, and suspect 2 has 1 band. And this is the sperm DNA that was found on the victim of the rape case. So you can see here that these two are a match. So suspect 1 was the person who committed the crime. So what we're doing is what's called restriction fragment length polymorphisms. We're pretty much breaking down DNA into fragments and sorting them according to size. We can use this in forensics, paternity testing, we don't know who the father is, detecting harmful diseases. The applications are amazing. And again, the fragments sort according to size, and the bigger fragments are on top and the smaller fragments are on the bottom. And we'll be doing this in class. So if you have any questions, please see me tomorrow in class. Thanks.